In our last discussion, we established this idea that an alignment is simply a statement of homology. That if we have two sequences of DNA that we can infer are descended from a common ancestor, that we can perform some sort of a algorithm to figure out exactly how they match up base pair by base pair to give us some inclination or some idea about what the ancestral sequence was like. Right? So homology is a statement of similarity due to ancestry. And so we do an alignment. In this case, it's a very simple alignment. We say there's been a deletion or an insertion, and we establish this idea that we may not know exactly which it is if we only have two sequences. And I asked you to start thinking about how we might be able to determine whether it's an insertion or a deletion. And the answer is if we gather additional sequences, particularly from closely related species, or perhaps species that are not as closely related but are maybe a close relative of these two. And what I mean by that is maybe we're looking at Drosophila sequences here. So we could add additional Drosophila sequences. Or we could potentially say, let's look at a different genus outside of Drosophila, and perhaps we could come up with a consensus or an idea about whether this T here represents an extra base pair that was gained as a mutation in sequence 1, or the loss of a base pair that was gained somewhere in the history of sequence 2. So we could even potentially, with enough data, come up with some ideas about the ancestral sequences based on these alignments. Now, these alignments are also critical for all sorts of other analyses that use these as the beginning data or a data set to do an analysis, including phylogenies, including population level analyses, and many others. Okay, so this is a simple pairwise alignment when we only have two. Now, in theory, there are infinite number of ways to align any two sequences, but at some point it becomes somewhat ridiculous. So if I want to, I could put all sorts of gaps here and then make that T line up with this T. But at some point, we get this kind of ridiculous scenario that is not very likely. And so the idea is to try and find the most likely alignment that gives us an idea of this shared ancestral connection between the different nucleotides in two DNA sequences. And so to do this, we need to account for both substitutions and gaps. Now, when mutations or changes and differences between the sequence are infrequent, this is rather trivial. It's pretty easy to do. We only have one or two substitutions and maybe one or two gaps here and there, and it does not become a very difficult task when the sequences are very, very similar. Okay? And so we could implement this in some sort of a mathematical way where we assign a cost represented here by these variables, where S is the cost of a substitution, W is the cost of a gap, and G is the cost of a gap opening penalty. And then we could implement this in some sort of equation to evaluate whether alignment A is least costly or better than alignment B or C or D, and maybe even do an arrangement, a wide range of many different alignments, and find the one that is best based on these costs. But there are multiple issues here going on that are not completely solvable. To some extent, even for a fairly simple alignment, this might be a somewhat of a complex idea about how we decide what the very best overall alignment. And that is especially true as sequences get more and more divergent. So how do I decide values for these parameters? Is a substitution worth or cost twice as much as a gap? Or is a gap twice as much as a substitution? How much when I extend that gap, make it two missing nucleotides or gained nucleotides rather than one, and so on. And the short answer is there is no a priori way to figure out a value for these parameters. And so even though we might have this fairly simple, straightforward method of saying, well, I assign costs to the different evolutionary events, and then I find the best alignment based on those costs, there's still some additional confounding factors here that can complicate the problem. But this is a critical common process that goes on all over the place. And in the next discussion, we'll look at a fairly straightforward alignment, although maybe a little bit more complex than the one we just looked at. And then we'll start talking about what happens as sequences get more and more divergent from one another. Now, alignment is a common process for both DNA sequencing data and it can be more and more problematic. So this is done when we're assembling genomes. It's basically an alignment issue. Now with genomes, it's not as much of an issue because we're sequencing the same parts of the genome over and over and over again, and we'd expect them to be identical with maybe some allelic variation, right, if we're looking at both copies of the genome, but very, very little variation compared to when we're looking at homologous sequences between different species. So the idea of aligning sequences that all come from the same genome is related, but not quite as complex as aligning sequences from different species, that especially as those sequences become more and more divergent. So how do we establish this homology? Well, we're simply looking at, do two DNA sequences share more similarity than we would expect by random chance? And so we need to establish how much similarity we would expect by random chance. And so we could take randomly selected pieces of DNA and just align them up to get some idea of how much similarity we would expect just by chance. And so we would get this kind of fairly narrow peak where just by chance we would have some level of similarity. And it's very unlikely that two randomly chosen pieces of DNA would be very, very similar. And also, although there's less of a tail here, it's very unlikely that they would be very, very different. Now, it turns out that this is a rather simple peak to talk about. And so most DNA sequences, just randomly chosen, are going to peak somewhere around 75% difference or about 25% similarity. And that's simply because of the laws of probability. There are only 4 nucleotides. So if I take two DNA sequences of the exact same length and line them up, we're going to have a 75% similarity, one of every 4 base pairs just by chance. And so at that point, we've kind of maxed out the amount of diversity that can be present in 2 DNA sequences and still do a meaningful alignment. Now, if we add gaps to that equation, we can get a similarity that is lower than 25% or differences higher than 75%, right? But at that point, it just becomes kind of silly to even put gaps into alignment because we can just say, well, let's just simply take out all gaps, align the sequences so that we have 25%, a little more, a little less. But at that point, there are so many DNA sequences, so many differences between DNA sequences that essentially it is a meaningless alignment. We've got a 25% similarity, but those don't really represent homologous positions at the DNA. They simply represent the laws of probability. And so we are looking for something between 100% similarity and 25% similarity, hopefully somewhat closer to 100% similarity, in order to get some meaningful information out of an alignment. Okay? And so here we have an example of what we call a local alignment, where we are allowed to infer that we're either missing data on either side or that there have been large mutations. Or we can maybe assume if we have some evidence and say that no, these pieces are exactly the same, and so we need to line them up as much as possible and do a global alignment. Okay, so target sequence, query sequence, we come up with these end results. And so depending on the constraints on a system, we might have different rules about what we allow for gaps versus substitutions. Okay, so a local sequence alignment tries to just find pieces that match. A global alignment is assuming that those two sequences have some sort of ancestral sequence that was identical and we're not missing any information, particularly on either side. Now, most of what we're going to be talking about and relying on for the rest of the semester are nucleotide alignments, DNA. However, we could potentially also take proteins where we have 20 different possibilities and do a protein alignment. And this is particularly important for protein coding sequences. And so we are now going to extend this idea of a pairwise alignment to a multiple sequence alignment. Now, I said in theory we have an infinite number of alignments even for just a pairwise alignment. But at some point it gets so ridiculous and our costs get so high that we really don't need to spend much time searching amongst these really ridiculous alignments. However, as we add more and more sequences, even if they are somewhat conserved, we may have more potential alignments that we need to consider with those scores. And so what we do is we sum the score of all of the pairwise alignments or some other measurement, and we'll look at some of those in our next discussion, but we sum all of these alignment costs together. And again, if there are relatively few changes, here's one at the amino acid level, it becomes fairly trivial. And so it's fairly obvious that even though some of these sequences have one amino acid at this position and other sequences have another one, that we should not put any gaps, right? So if we wanted to, we could put a gap in and make all the D's line up, put another gap in and make all the N's line up. And then I think there's some difference. There's some E's down there too. But that becomes somewhat ridiculous and not a very likely alignment. And so if we're in that kind of, 100% to 90% similarity range, then the alignment is rather trivial. And no matter what kind of score we use or what sort of algorithm we use to align them, we'll get very, very similar end results. Okay, but as they become different, the amount of effort and the amount of alignments that we might need to evaluate grows exponentially. So every time I add a sequence, I have an exponential number of different combinations that I might need to evaluate and check out the score in order to have some idea that I have the very best alignment overall. Okay, and so this is also an NP-complete problem. Now, an NP-complete problem means a problem for which we don't ever know 100% if we have the correct solution, and the number of possible solutions that we may need to evaluate grows exponentially. So rather quickly, if we just have 20 or 30 sequences, if we wanted to evaluate every possible phylogeny, and even if we limited that to every possible reasonable, I said phylogeny, but we're talking about alignments. If we wanted to evaluate every possible alignment, or even to kind of scale that back, every possible reasonable alignment, and that might pose another problem, is how do we define what a reasonable alignment is? Where do we draw that line? Even then, we may need to evaluate trillions upon trillions of different alignments, and it might be impossible to do a complete search. And so for NP-complete problems, we usually are left with doing some sort of a partial search, but in a manner that means that we've reasonably covered the entire search landscape. And we'll talk about this more when we talk about phylogenies and how we find the best phylogeny, because that also is an NP-complete problem. And so realize there's a little bit of a catch-22 here. We're doing these alignments so that we can find the best overall phylogeny. Right? That's one of the reasons. There are other reasons also, but much of what we're going to look at in this first unit is phylogenetic theory and reconstruction. So we make an alignment to find the very best phylogeny, but knowing something about relationships, about the phylogeny, might help us actually to make a better alignment. So there's that kind of paradox or catch-22. And we'll look at some approaches and ways that we can, at least in a partial method, resolve or at least alleviate some of the impact of that paradox.